In this video we will discuss about some of the observations that researchers has observed when they use their hydrogels, newly developed hydrogel, to activate the T cells via anti-CD3 proteins. So some of the things that they are expecting is F-70 phosphorylations and the movement of this F-actin that we have covered briefly in the previous video. In this video we will discuss those results and their impact or their changes with the different stiffness of the hydrogels. So first what they did is they checked whether the hydrogels are developed properly or not. So they checked the anti-CD3 are properly printed on their hydrogels, and then they did it with this DAPI. They check whether the cells are bind to the anti-CD3s or somewhere else. Thereafter they check whether the ZAP70 phosphorylations is occurred or not using anti-ZAP70 phosphorylations. And then they check the phalloidine to check for staining the this F-actins, and thereafter they overlay the whole picture without DAPI, and here we can see that the ZAP70s are mainly expressed within the cells. The quantifications of these images are shown in this panel B and C, where we can see that in panel B what they did is they presented this ZAP70 phosphorylations intensity, and what they find out is that with increase in the stiffness the ZAP70 phosphorylations intensity increases. So, and this is also in line with the observations that are reported in the previous literatures, and this also makes sense as more stiffer the hydrogel is, then it will cause more mechanical response to the cells, and thereby the higher stiffness will cause more ZAP70 phosphorylations, or it triggers these T cells activations more. And in terms of the ZAP70 centralizations, that is whether it is expressed on the plasma membrane, near the plasma membrane, or at the center. When they check it with the different stiffness of the hydrogels, they find out that there is not much difference in the ZAP70 centralizations in different stiffness of the hydrogels, because and their results finally conclude that the stiffness does not have much impact on the movement of the ZAP70 phosphorylations. After that, they wanted to check the F-actins movement. It has been reported in the literatures before the publication of this paper is that during the T cells activations, F-actins first move to the plasma membrane, and then it will move to the, and thereafter it will move to the center of the cells. So doing early activations, they check the F-actin movements, and these results are similar to what we have seen in the previous video, but here they have did some more quantifications, and they did their experiments on patterned printing of anti-CD3 cell and unpatterned printing of anti-CD3 cells. And unpatterned printing of, and these comparisons is shown in the supplemental figures. Here we will discuss the results that are presented here. So they have checked ZAP70 phosphorylations and F-actins, and they are able to see that F-actins are mainly expressed at the periphery or near the plasma membrane for this 2 kilopascals, and this movement of F-actins towards plasma membrane increases with increase in the stiffness of the cells. That can be seen in the panel right hand side of the panel A of the right hand side figure, that with increase in the stiffness, the more the peaks towards the periphery of the cells increases. And when they quantified in terms of the spreading area, they are able to find out that the spreading areas with increase in the stiffness, the spreading areas of the cells increases, and it is statistically significant compared to the spreading areas with 2 kilopascal. And the, from this one can easily make a understanding that when the cells are spread apart, their receptors are more exposed to their respective proteins, so it will cause more activations. So this will further complement why we see more ZEP70 phosphorylations, because it has more spreading area, and T cells are more exposed to the their receptor proteins. When it comes to the F-actin clearance, they see that F-actin clearance is high in terms of in the 2 kilopascals, but it is low for 12 kilopascals and 50 kilopascal. So this also makes sense, as most of the F-actins are moved towards the periphery of the cells with increase in the stiffness. So its clearance at the center will be higher. At the center will be lower compared to the this F-actin clearance at 2 kilopascal or low stiffer cells. And when they check the ZEP70 phosphorylations again, they are able to see that the ZEP70 phosphorylations with increase in the with increase in stiffness, they see more. With increase in stiffness, they see more ZEP70 phosphorylation signals. One more thing that I want to clarify: the figure on the right hand side is for the unpatterned anti-CD3 printing on the hydrogels, and left hand side figure is for the patterns anti-CD3 proteins printed on the hydrogels, and the comparisons are shown in the supplemental figures. Here we are discussing the results individually, but we can see that the results seems to be similar for this ZEP70 phosphorylations intensity. And when they check the ZEP70 centralizations, they do not see any much difference in the unpatterned printing of the anti-CD3 on hydrogels conditions case. In that case, so from this they conclude that there is not much difference that has been observed with the pattern anti-CD3 printed on the hydrogels on unpatterned anti-CD3 printed on the hydrogels. Only changes that they have observed is about the with increase in stiffness they see more spreading area, and that could means that the cell receptors are exposed to their respective proteins more compared to the lower stiffer conditions. That will cause more activations of the cells or more ZEP70 phosphorylations. So I hope you understood the comparisons between pattern anti-CD3 protein printed on the hydrogels and unpatterned anti-CD3 protein printed on the hydrogels. If you need to understand more about these results and wanted to see the comparisons between those results, you can go through the supplemental figures that has been you can go to the supplemental figures of this paper, and the reference link of the paper has been provided in the description. That's all for this video. Thank you for your time.